Hi guys, let's start our chapter number 6, Plant Nutrition. Overview, what we are going to learn in this. Key learning objectives. Understand photosynthesis and its fundamental equation. Explore how plants use carbohydrates. Investigate leaf structure and photosynthetic adaptations. Examine critical factors affecting photosynthesis. Alright, so solar energy and fuel production. Global energy challenges. Growing energy demand, increasing human population requires more fuel. Current energy sources. Fossil fuel dominate energy production. Renewable sources like wind and solar gaining traction. Environmental concerns. Fossil fuel combustion increases atmospheric carbon dioxide. It contributes significantly to global warming. Plant-inspired energy solutions. Number 1 is natural photosynthesis. Plant converts sunlight into usable energy, which is carbohydrate. Carbon dioxide is consumed and not produced. Biofuels consideration. Growing crops for fuel competes with food production. Potential habitat disruption. Next we will see about leaf structure and photosynthetic tissues. Let's see leaf anatomy layers. First layer is upper epidermis. Characteristics is they are tightly packed cells. Their primary function is water retention and light transmission. Next layer we have palisade mesophyll. The characteristic of these cells are that they are tall and as well as narrow cells there. Its primary function is intense photosynthesis. Next we have spongy mesophyll, rounded loosely packed cells, gas exchange, moderate photosynthesis is their function. In lower epidermis, similar to upper epidermis, and function is minimal water protection. Now let's see the functions of tissues in detail. Upper epidermis, tightly packed cells without chloroplast, secretes waxy cuticle for waterproofing, allows maximum light penetration. Next we have palisade mesophyll, tall narrow cells, highest chloroplast concentration, optimal sunlight absorption, primary photosynthetic layer. Spongy mesophills, let's see what they do. They are loosely arranged cells. Fewer chloroplasts are present as compared to palisade layer. They are critical for gas exchange. In there, carbon dioxide diffusion taken place, oxygen circulation taken place, and water vapor movement also is happening there. Next we have cellular components. First is chloroplast. So energy conversion organelles. Chloroplasts are the energy conversion organelles. Cell wall, they are for structural support. Vacuoles, they store and cellular maintenance. Nucleus, this is genetic control center. Photosynthetic factors, key requirements: chlorophyll, sunlight, carbon dioxide, and an appropriate temperature. Limiting factors, conditions that restrict photosynthetic rate, interplay between light intensity, carbon dioxide concentration, and temperature. So, in photosynthesis, let's talk about leaf structure and photosynthesis. Carbon dioxide enters through stomata from the atmosphere. Water travels from roots via xylem vessels. Materials diffuses through air space into cells. Chloroplasts are the synthetic photosynthesis powerhouse. Alright. Location of photosynthesis is inside chloroplast. Distribution most concentrated in palisade cells. Can move around to maximize sunlight exposure. Chloroplast structure. It contains membrane, cytoplasm, starch grain, and chlorophyll containing membrane stacks. Leaf adaptations for photosynthesis includes their large surface area, which helps in maximizing light absorption. Then its thin structure, it facilitates efficient gas exchange. Palisade mesophyll cells, what they do in the leaf, packed with chloroplast, positioned to capture maximum sunlight. Some experimental skills for investigating photosynthesis. So, testing of leaf for starch, for example, purpose is to determine if photosynthesis has occurred or not. We will take the key equipment: beaker, burner, iodine solution, alcohol, fresh green leaf. Learn these experimental techniques, equipment, name of all the important processes, as they are very important for our exam questions. Procedure: boil leaf to break cell membrane, remove chlorophyll using alcohol, apply iodine solution, and black blue color will indicate that there is an starch presence in it. Alright, next, chlorophyll rows. So what is the role of chlorophyll? Method: we will use variegated leaf. Observation: starch only present in green chlorophyll containing areas. Light density technique: block light from part of the leaf using black paper. Goal: demonstrate light requirement for photosynthesis. Next, carbon dioxide roles. The setup should be 2 transparent containers, one with potassium hydroxide which removes carbon dioxide, and then one with distilled water. Factor affecting photosynthesis rate. So these are: number 1, environmental influences, very important. Raw material supply, carbon dioxide and water. Sunlight quantity provides energy for reaction. Temperature affects enzyme activity. And then chlorophyll quantity determines photosynthesis speed. Alright, the controls in the experiment would be: purpose, ensure accurate comparison. Alright, keep some points in your mind as it comes in the exam. If they ask you, write down some of the control points from experiment, so you can write. Maybe they will ask, write few steps or methods to perform this test, so you can write. Alright, so here experimental control: ensure accurate comparison. So examples: unblocked leaf section in light experiment, green leaf parts in chlorophyll investigation. What are the key experimental principles? Always include control group, minimize variable, make careful observation, and draw precise conclusion. Okay. Next is safety consideration. Alcohol handling, it is flammable, so be very careful. Collect only when ready to use. Use damp cloth if fire occurs. Now, photosynthesis process, our key takeaway are: photosynthesis is a complex process, multiple factor influences efficiency. Careful experimental design is crucial for understanding. Alright, overview of the process of photosynthesis. A process where plant convert light energy into chemical energy. The equation is 6CO2 carbon dioxide plus 6 water molecules gives us glucose and oxygen. Key components of photosynthesis: chlorophyll. Where is the location of chlorophyll? It is located in chloroplast. What is the function? It transfers light energy into chemical energy. It is essential for converting solar energy into usable plant energy. Important to learn, please memorize. Next, glucose production and uses. Alright, primary product of photosynthesis is glucose. Versatile application: it's energy storage, converted to starch, cell wall construction, cellulose, transport sugar, sucrose, protein synthesis, all is happening due to glucose production. And chlorophyll production. Okay, leaf structure and photosynthesis. What is the leaf characteristics now, and what is its role in photosynthesis? So leaf characteristics: large surface area, maximize sunlight capture, carbon dioxide absorption. The key features of stomata: it is located on lower leaf surface, allows carbon dioxide diffusion and control gas exchange. Now, limiting factors in photosynthesis. Types of limiting factors: light intensity. no photosynthesis in darkness, slow in dim light, and maximize rate reach at optimal light intensity. Carbon dioxide concentration, it increases CO2, increases photosynthesis rate plateaus at maximum concentration. The third one is temperature. Chemical reactions slow at low temperature. Enzyme catalyzed reaction affected. Optimal temperature varies by plant. Alright, next is limitation factor visualization experimental method measuring photosynthesis rate. So we have an aquatic plant technique. We can do the bubble counting, gas volume measurement, and hydrogen carbonate indicator, which shows us the color changes. Experimental variables. So we have independent variable and control variable. In independent variables, we have light intensity, carbon dioxide concentration, and temperature. In control variables, we have plant size, water conditions, and experimental setup. Starch testing procedure. Alright, what we have to do, step number 1, boil the leaf to break cell membrane, remove chlorophyll with alcohol, apply iodine solution, observe color change, and then use control sample for comparison. Now, practical consideration, let's talk about stomatal behavior. Close during extreme heat and the bright conditions, prevents excessive water loss, reduces photosynthesis efficiency. Guys, these all are very important points. You have to learn all of them in order to perform well in the exams. Okay, understanding is good, but you have to produce the answer in the exam, for which learning and memorizing is required as well. Advanced insights. Photosynthesis rate depends on multiple interacting factors such as environmental conditions significantly impact plant productivity. And we also need to understand about limiting factors, which is crucial for an optimization. Alright, now research and application, glasshouse design consideration. Okay, so we have to optimize growing condition, as mentioned here, that we have, it is crucial for agricultural optimization. So we can use glasshouse design consideration, which can optimize growing condition, where we can control environmental factors and maximize photosynthetic efficiency. So our key takeaways here is photosynthesis is a complex multi-factor process. Environmental conditions critically influence plant productivity, such as control of light, carbon dioxide, temperature, precision. These all maximize plant growth. Alright, now let's talk about photosynthesis and plant cells. Alright, so fundamental, as we know, chemical equation, as we know the breakdown of this molecular equation. Carbon dioxide, 1 carbon atom, 2 oxygen, glucose, 6 carbon, 12 hydrogen, and 6 oxygen atoms. Key photosynthesis components are sunlight, chlorophyll, and enzymes. Alright, now leaf structure and cell types. Leaf tissue adaptation. Here we will talk about palisade mesophyll cell. Here the highest number of chloroplasts found, optimized for photosynthesis, densely packed with chlorophyll. Now chloroplast distribution, cell type and mean chloroplast per cell. In palisade mesophyll we have 90 prox, spongy mesophyll we have around 50 to 60, guard cells we have fewer than spongy one, and in the upper epidermis we have minimal or none chloroplast. Alright, chlorophyll function. It captures light, it enables photosynthetic reaction, it converts light into chemical energy. Carbohydrate synthesis and usage. Carbohydrates production. Synthesized during photosynthesis, glucose is primary product. Carbohydrate uses is energy storage, structural components, metabolic processes, and growth and development. What are the photosynthetic factor? Temperature effect, limiting factor. Temperature is a limiting factor. Carbon dioxide concentration, light intensity, temperature, enzyme efficiency. These all are the limiting factors. Alright, experimental insights. Photosynthesis rate measurement. So we can observe how much oxygen volume released, what are the temperature variations, then carbon dioxide concentration effects. Now, ecological significance of this: plants are the primary energy producer. Animal dependent plant generated energy, fundamental to global ecosystem functioning. Our key takeaways here are: photosynthesis is a complex enzyme driven process. Chloroplasts are critical for energy conversion. Environmental conditions significantly impact photosynthetic efficiency. Alright, now let's talk about photosynthesis and plant carbohydrate utilization. So, molecular modeling of photosynthesis. Egg cotton molecule modeling technique. Here objective is create a hand-on model to understand photosynthesis. Some material that we are going to use is egg cartons, color pans, small balls representing atoms. Now, atom labeling, you can use oxygen 18 red balls, carbon 16 black balls, and hydrogen with 12 blue balls. Now, construction steps: create water molecules H2O, construct carbon dioxide molecules, build glucose molecule using a specific egg carton configuration, optimal deconstruct and reconstruct for practice. Key modeling insight: reflection, evaluate models' effectiveness in understanding photosynthesis, analyze how the model represents chemical transformation. Now, let's talk about plant carbohydrate utilization. Glucose production and utilization. Primary products of photosynthesis includes glucose and oxygen. Energy and store mechanism: energy release, number 1, and energy store in the form of starch. So, energy release: glucose used for cellular activity, supports active transport, enables protein synthesis, energy release through respiration. When you talk about storage strategy, starch, you will talk about it converts excess glucose into starch. Then starch characteristic is they are not water soluble, they does not affect cellular osmosis, easily convertible back into glucose. Alright, next is glucose transformation pathway. You have sucrose production, how glucose converted for plant-wide transportation, and then they are transported via phloem tubes. In glucose transformation pathway, the second thing we have is cellulose formation. It builds cell wall, different molecular linking compared to starch. Third is nectar creation, attracts pollinators, contains various sugars, supports reproductive strategies. Fourth, we have amino acid and protein synthesis, requires additional nitrogen from nitrate ions, enables protein production, critical for plant growth. Now, let's talk about chlorophyll production, requires nitrogen and magnesium. Then deficiency leads to yellowing leaves and reduced photosynthetic capacity. Next, we have leaf structure and photosynthesis. Leaf anatomy components: it has lamina, that is the broad flat structure, surface of the leaf. Next, we have leaf stalk, vascular bundles, and veins. Photosynthesis optimization: structural adaptations take large surface area, thin structure, extensive vein network. Now, vein configuration, if you see, it has dicot plant network like vein arrangement, monocot plants parallel vein arrangement. So, you can see the difference between dicot plants and monocot plants. Next, we will talk about cellular photosynthesis occurs in chloroplast. Chlorophyll spread on membranes. It requires carbon dioxide, water, sunlight. Mineral deficiency impact table. Alright. Now, we will talk about two elements, nitrogen, magnesium. So, in mineral salt, it is nitrate ion. The purpose is protein synthesis. Deficiency can cause weak growth and yellowing of leaves. Second element is magnesium. Its mineral salt is magnesium ion. The purpose is chlorophyll production. Leaf yellowing, reduce photosynthesis if deficiency occurs then. So, the key takeaways for us is photosynthesis is a complex multi-step process. Plants have sophisticated mechanism for energy production and utilization. Mineral balance is crucial for plant health and growth. So, that's it for this chapter. Hope to see you in our next chapter once we continue with our next chapter, which is chapter number 7. See you soon.